Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing DBT BET JRF question paper, and I will be discussing the part 2 of the 2021 conducted exam. Part 1 has already been discussed. So coming to the next question. The consequences of DNA base change in a mutation are maximum if the base change is located in the. So, if suppose a base substitution occurs in the third position of the codon, there is a good chance that synonymous codon is generated. That is, synonymous codon means different codons that encode the same amino acid are known as synonymous codons. Because third position is the wobble position. So, the consequence of DNA base change in a mutation are maximum at first and second position of a codon. Answer is option 1. The telomerase enzyme is a reverse transcriptase. Answer is option 3. Though DNA and RNA are nucleic acids, isolating RNA in the laboratory requires extreme precautions and pre-preparations than isolating DNA. This could be because because RNA is more prone to hydrolysis than DNA, and because ribose sugar in RNA has a hydroxyl group at the 2' position, making it susceptible to hydrolysis. So RNA is more prone to hydrolysis than DNA. Answer is option 3. Type 2p restriction endonucleases will always recognize palindromic sequences. Answer is option 3. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. So assertion A, ionizing radiation can cause damage to DNA. Ionizing radiation generate free radicals. In the light of the above statement, choose the most appropriate answer. So both A and R are true, and R is the correct explanation of A. Answer is option 1. Then the lysosomal lumen is maintained at an acidic pH by H plus ATPase that pumps H positive ions into lysosome. Answer is option 1. Then which one of the following is an example of nuclease? It is DNase 1. So a nuclease is an enzyme capable of cleaving the phosphodiester bond between the nucleotides of nucleic acids. So it is DNase 1 is a nuclease. Then next one. Which one of the following statement is not common between oxidative phosphorylation and photophosphorylation? Generation of ATP, yes. Involvement of electron transport, yes. Involvement of kinase, no. Involvement of oxygen, yes. So the answer is option 3. Which one of the following combinations signifies similar function? So cryptochromes. Cryptochromes are class of flavoproteins found in plants and animals that are sensitive to blue light. Then cytochrome. Cytochrome are iron heme containing a heme with central iron atom at its core, and they are involved in electron transport chain. Then phytochromes are a class of photoreceptors in plants used to detect light. And fluorochrome. Fluorescent dyes are fluorochromes that are used in biological staining to produce fluorescence in a specimen. So which of the combinations signify similar function is that of cryptochrome and phytochrome. They both are photosensory receptors. Answer is option 2. Cryptochrome, phytochrome. Then next one. E-class homeotic genes in Arabidopsis thaliana are involved in the formation of. So according to the ABC model of flower development, A and E class protein complex develop sepals in the first wall. A, B and E develops petals. B, C and E helps in stamens in the third wall, and C and E specify carpels And D class proteins interact with E class to specify ovule identity genes. So E class genes are involved in the identity determination of sepal, petal, stamen, carpels. So as per the answer key, petals and carpels are given as the correct answer. And as per me, not only 3 but 1 is also correct. Sepal and petals can also be taken as correct answer. Anyway, if you have any comments, you can give it in the comment section. Then next one. When the critical night length in winters is disrupted by a pulse of red light followed by a pulse of far red light, short day plants will flower, long day plants will not flower, long day plants will flower, short day plants will not flower, both long day, short day will flower, none of the plants will flower. Okay, long day plants, they require periodic exposure of light exceeding the critical period to induce flowering. And short day plants, they require the periodic exposure of light less than the critical period to induce flowering. Now, when critical period is interrupted by red light, long day plants flower. See here, this diagram. So when critical length period is interrupted by red light, long day plants will flower, short day plants will not flower. When critical period is interrupted by far red light, short day plants flower. Now if it is far red light alone, short day plants will flower, long day will not flower. And when plants are exposed to red and far red light in alternation, the final treatment determines the effect. So, when the critical night length in winters is disrupted by a pulse of red light followed by a pulse of far red light. So, we have to take the effect, the final treatment. So, final treatment was with far red light. So, here short day plants, they will flower, whereas long day plants will not flower. Answer is option 1. Then next one. A plant species nearing its extinction due to viral infection has been chosen for micropropagation by tissue culture. Which explants will be the most appropriate to produce virus-free plants? So naturally, the meristem region is more suitable for propagation of virus-free plants. Answer is option A, that is A only, shoot apical meristem. Then next one. The rice blast fungus Magna porthae invades rice plants in a manner typical of many foliar pathogens by producing specialized infection structures called as apressoria. Answer is option 2. The first alkaloid to be isolated and characterized from plant is morphine. Answer is option 1. Then next one. Seeds of Arabidopsis thaliana placed on Murashki and Skoog media without any hormones germinate faster than in the medium that contains abscisic acid. Inhibits seed germination. So seeds of Arabidopsis thaliana placed on Murashki Skoog medium without any hormone germinate faster than in the medium that contain abscisic acid because abscisic acid inhibits seed germination. Next one. Which one of the following is a non-climatic fruit? So a non-climatic fruits once harvested do not ripen further. They produce very small amount of ethylene and do not respond to ethylene treatment. There is no characteristic increased rate of respiration nor production of carbon dioxide. Non-climatic in which respiration shows no dramatic change and ethylene production remains at very low level. So which of the following is a non-climatic fruit? It is wild strawberry. Answer is option 3 out of these options. Then next one. Which one of the following statements are true for gibberellins? Gibberellins promote seed germination. Yes, gibberellin cannot stimulate leaf growth. No, that is wrong. Gibberellin cannot stimulate stem growth. That is also wrong. Gibberellins can be exogenously used to increase plant growth. Yes. Gibberellic acid 3 is predominantly used in agronomic and horticultural practices. Yes. So A, D and E are correct. Answer is option 2. Then which one of the following is not a secondary metabolite? Acetyl-CoA. It is not a secondary metabolite. Then next one. Which one of the following classes of compounds is generally accumulated as an anti-herbivore response in plants? So it is alkaloids. Answer is option 2. Which one of these polysaccharides is induced after pathogen or microbial attack? It is callose. So callose is a polysaccharide in the form of beta-1,4-glucans with some beta-1,6 branches, and it exists in the cell wall of a wide variety of higher plants. So the answer is callose. Then GA2 oxidase gene from bean is overexpressed in a wheat plant by genetic engineering. Which one of the following phenotypes correctly describe the resultant transgenic plant? So GA2 oxidase, it catalyzes the catabolism, namely the conversion of bioactive gibberellic acids and their immediate precursors into biologically inactive gibberellic acids by 2-beta hydroxylation. So what will happen? Now the GA2 oxidase gene for bean is overexpressed in wheat plant. So GA2 oxidase gene, it is having a negative effect on or it inactivates the gibberellic acids. So naturally there will not be any internode elongation. So the plant will be shorter than the wild type. Answer is option 1. Then next one. The tDNA of agrobacterium must be cut out from its circular plasmid for its transfer into plant cells. Which one of the following Vir proteins are involved in this process? So it is Vir D1 and D2 that are involved. Then root initiate from the pericycle. Answer is option 2. Which one of the following is not a characteristic feature of scotomorphogenetic development? So scotomorphogenesis is development of a seedling in the dark. In scotomorphogenesis, the seedling is characterized by long, thin hypocotyle. So first option, long hypocotyles, is correct. Folded cotyledons, so closed cotyledons, that is also correct. Option 3. Which protect the shoot apical meristem, closed apical hook that protects unfolded cotyledons. So option 2, apical hook, is also a characteristic feature of scotomorphogenetic development. So which is not a characteristic feature? Expanded leaves. Answer is option 4. Then next one. Which one of the following can be used as a selection marker for developing transgenic plants? So hygromycin phosphotransferase can be used. GFP, beta galactosidase, beta glucuronidase, all these are reporter genes. They have asked for selectable marker genes. So it is hygromycin phosphotransferase. Then next one. Which one of the following treatment is required for flowering in a winter annual type of Arabidopsis plant? So in winter annuals, high level of the floral inhibitor FLC, that is flowering locus C, suppresses flowering prior to vernalization. And vernalization is the induction of plants' flowering process by exposure to prolonged cold of winter. So which of the following treatment is required for flowering in winter annual types of Arabidopsis plant? It is vernalization treatment, that is prolonged cold period. Answer is option 1. Next one. Which one of the following statements are true regarding specialized embryonic structures peculiar to the grass family? The cotyledon have been modified by evolution to form an absorptive organ called coleoptile. The epicotyle give rise to stem and leaves and is covered by a protective sheath, that is the coleoptile. Then scutellum forms the interface between embryo and starchy endosperm tissue. That is correct. Because embryos of monocotyledons possess only one cotyledon and the cotyledon is called as scutellum. Then next one. Coleoptile covers and protects the first leaf while buried beneath the soil. That is also correct. The base of the hypocotyle has elongated to form a protective sheath around the radicle called the scutellum. That is not correct. So the answer is B and C only. Answer is option 3. Then 71 to 90, those questions are from fermentation technology and I am not well good with that subject, so I have not answered it. Okay, next question. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. Gene sequences are aligned using identity matrices instead of substitution matrices. The 4 bases in DNA cannot be replaced with each other. So there are two types of matrices, that is identity and substitution matrix. Identity matrix only assign points to residues that are identical, whereas substitution may assign points to residues that are different yet alike in conservative properties. That is, they may be having the same physiochemical properties. That is in the case of amino acids. So here gene sequences are aligned using identity matrices instead of substitution matrices, and the 4 bases in the DNA cannot be replaced with each other. So that is correct. So both A and R are true and R is the correct explanation of A. Then, which one of the statements relating to properties and structures of two proteins is most appropriate? So, two proteins with very similar secondary structures will have similar tertiary structures. Two proteins with very similar secondary structure will have similar stability. Two proteins with very similar tertiary structure will be localized to similar compartments inside the cell. Two proteins with very simple tertiary structures will have very similar secondary structures. It is not simple, it is similar. Answer is option 4. Two proteins with very similar tertiary structures will have similar secondary structures. Because the overall packing and arrangement of secondary structures from the tertiary structure of a protein. Sorry, not from, it is from the tertiary structure of a protein. The overall packing and arrangement of secondary structures form the tertiary structure of a protein, and secondary structure prediction is an intermediate step in tertiary structure prediction, as in the case of threading analysis, and close protein homologs should adopt the same secondary and tertiary structures. Then next one, match the components in list 1 with list 2. Sequence alignment, BLAST, structural alignment, DALI, fold prediction, Rosetta, and review of literature, PubMed. So answer is option 2, A2, B4, C3, D1. Then next one. The TRP fluorescence can be used to study protein folding and unfolding. What properties of the TRP are critical in ensuring that this can be used for studying the process? TRP is an environment sensitive fluorophore which is typically buried in a folded protein. TRP is a positively charged amino acid. TRP is the only amino acid present in most of the proteins, and TRP is better hydrogen bond acceptor than most other amino acids. Answer is option A, that is TRP is an environment sensitive fluorophore which is typically buried in a folded protein. Then next one, given below are two statements. Peptide bond is a planar bond. The Ramachandran plot describes omega torsion angles in proteins. In the light of the above statement, choose the most appropriate answer from the options given below. Both statement 1 and 2 are correct. Okay, wait. The peptide bond is a planar bond. The Ramachandran plot describes omega torsion angles in protein. Ramachandran plot describes phi and psi angles in proteins. So statement 1 is correct, whereas statement 2 is incorrect. So answer is option 3. Statement 1 is correct and statement 2 is incorrect. Next one. Protein folding is highly cooperative. Which one of the following statement defines this cooperativity? So the answer here is the protein chain completely unfolds if key interactions are broken in an all or none manner. So if you plot a protein disrupting factor, that is like high temperature, against folded fraction of the population of protein, you will get a sigmoidal curve that shows highly cooperative curve. Then at the curve's midpoint, half of the population is folded and half is unfolded. And the population of partially folded intermediates is not at all present, or it is very, very negligible. So all or none means proteins are either folded or unfolded with negligible population of partially folded intermediates. So the answer here is option 4, that is the protein chain completely unfolds if key interactions are broken in an all or none manner. Then next one. Given below are two statements. Serine is a part of a catalytic triad in proteases that also include histidine and aspartic acid. Yes, catalytic triads are responsible for peptide hydrolysis. I have already discussed a similar question to this in part 1 with serine proteases. So the answer here is both statement 1 and 2 are correct. If two sequences are 1 PAM apart, they will be. So 1 PAM means 1% change in overall residues, or 1 point accepted mutation per 100 residues. That means 99% will have identical amino acid residues. So the answer is option 1. Next one. The contact map distance matrix of a protein provides a two-dimensional view of a three-dimensional structure of a protein. How can you obtain a proper three-dimensional structure from the contact map? So contact map, they provide a more reduced representation of protein structure than its full 3D atomic coordinates. So using contact map along with computational modeling techniques, we can obtain a proper three-dimensional structure of the protein. So answer here is option 2, using the contact map along with computational modeling techniques like simulated annealing. Answer is option 2. Then next one. Which one of the following is true about phylograms and cladograms? So phylogram is a branching diagram that is assumed to be an estimate of phylogeny. The branch length are proportional to the amount of inferred evolutionary change. A cladogram is also a branching diagram and assumed to estimate of phylogeny, where branches are of equal length. So phylograms show common ancestry but not time. They show time also. Branches of cladograms are proportional to the evolutionary time. It is not cladograms but of phylograms, the branches are proportional to evolutionary time. Cladograms show common ancestry but not time. That is correct. There is no difference between cladograms and phylograms. There is difference between cladograms and phylograms. Then next one. Two sequences PLAVAV and PLLV are aligned using Needleman-Wunsch algorithm with scores. Match is equal to 1, mismatch is -1, gap initiation is -1, and gap elongation is -2. The alignment with the best score according to this algorithm will be. So the answer here is option 2. How to solve this algorithm? I have given a description I have given a link in the description box. You can refer to that link. It is also a YouTube channel, but the algorithm has been very beautifully explained. How to calculate all these things based on that, and this is how I got it. So gap penalty you have to calculate. They have given gap initiation and gap elongation. So gap initiation plus gap elongation, that is -1 plus -2, it will be -3. That is how you calculate gap penalty for this particular question. Okay, so you can refer to the link in the description box and you can do and see this particular algorithm. Then next one. A protein sequence was isolated from a novel source. During an initial sequence similarity search through BLAST, no homologous sequence was identified. A further BLAST search should be run by changing the score matrix to. So it is PAM 250. Because no homologous sequence was identified, so PAM 250 is used. The in vitro ADMET analysis cannot provide information about. So ADMET, it stands for absorption, distribution, metabolism, excretion and toxicity. And the predictions of the ADMET properties play an important role in the drug designing process. That is absorption, that is how much of the drug is absorbed and how quickly, that is bioavailability of that particular drug. Distribution, where is the drug distributed within the body, what is the rate and extent of the distribution. Metabolism, how fast is the drug metabolized, what is the mechanism of action, what metabolite is formed and is it active or toxic. Then comes elimination, how is the drug excreted and how quickly. And then comes toxicity, does this drug have toxic effect to body system or organs. That is in the case of ADMET. Now they have asked for in vitro ADMET analysis. So blood-brain barrier permeability. In vitro models of the blood-brain have been developed to study permeation mechanism. Then comes option 3, that is metabolic stability. That is their ADMET, where M is metabolism. So naturally metabolic stability is also involved in vitro ADMET analysis. Cytochrome P450 inhibition. In vitro studies about cytochrome P450 inhibition are also available for in vitro ADMET analysis. Adverse reaction to drug, that is not available. That information is not provided. So the answer is option 2. Then K-tuple method is associated with dynamic programming. Answer is option 2. The motif D, square bracket TS, X, 2, curly brackets GH and L motif will have sequence. So square brackets TS means either T or S has to be present. X, any two amino acid, X2, any two amino acids can be present. Curly brackets means they are not accepted at this particular given position. These amino acids are not allowed. So if you see, answer is option 1. But before that I will explain 2, 3 and 4. So if you see 2, 1 minute. Okay, if you see 2, D is okay, S okay, fine. Out of T and S, S has to be either S or T can be present. Okay, X2, so it can be any amino acid. G, the G should not have come at this position because it is given in the curly bracket. So G and H are not allowed at this position. So 2 is not correct. C for the third option. D, T, R, H should not have come at this position. H is not allowed at this position. Then 4, if you see D, R, instead of T and S they have given R. So that itself is not correct. It should be either T or S at this position, not any other amino acid. So option 1 is correct. D, S, A, R, R, L. Option 1 is Correct. Then next one. How many atomic positions are required to measure a dihedral angle? So a dihedral angle is defined by 4 sequentially bonded atoms. So the answer is option 3. Which one of the following will be used to assess structural similarity of biomolecules? So it is root mean square deviation. Answer is option 3. Then a profile can be generated from a multiple sequence alignment by obtaining position specific preference or probability of each amino acid. This can be used to identify homologs. However, the key difference between a profile alignment like this and hidden Markov model is hidden Markov model has the option to introduce gaps with position specific gap penalties. That is correct. Answer is option 1. Then next one. You have purified a protein X and observed the following. When run on a native page, it gives rise to a single band. When run on a non-reducing SDS page, you obtain two bands corresponding to 40 kDa and 60 kDa. When run on a reducing SDS page, you get three bands corresponding to 60 kDa and 30 and 40, 10 kilodaltons. What can you conclude about the purified protein X? Okay. In native page, no bonds are broken. When run on non-reducing SDS page, you got two bands 40 and 60. And when run on reducing page, SDS page, you got the disulfide bonds broken in the 40 kDa subunit, resulting in 30 and 10 kDa subunit. So totally there are three polypeptide chains or subunits, that is 30, 10 and 60. And the 30 and 10 are linked to 60 by weak interaction which got broken when run on non-reducing SDS page. So the answer here is option 2. X contains at least 3 polypeptide chains that form a complex. Then next one. What types of bonds generally stabilize antigen-antibody interaction? So they are weak hydrogen bonds and van der Waals forces. Then Tay-Sachs diseases. Tay-Sachs diseases autosomal recessive genetic disorder. Answer is option 3. Next one. Dysbiosis is a term associated with. So dysbiosis, it is defined as an imbalance in the gut microbial community that is associated with a disease. So it is associated with microbiome. Answer is option 3. Then next one. Single variable fragment or fusion protein composed of. So a single chain variable fragment is not actually a fragment of an antibody but instead is a fusion protein of the variable region of the heavy and light chain of immunoglobulins connected with short linker peptide of 10 to about 25 amino acids. So the answer here is VH plus VL joined by a flexible linker. Then next one. Human embryonic stem cells can be obtained from. So they can be obtained from inner cell mass of blastocyst. Answer is option 3. Then which gene is often been inserted in an adenoviral vector to treat cancer by suicide gene therapy? So suicide gene therapy is a therapeutic strategy in which cell suicide inducing transgene are introduced into cancer cell. So which gene is often being inserted in the adenoviral vector to treat cancer by suicide gene therapy is HSV-TK. Answer is option 1. Then next one. Which one of the following is the most common adjuvant composed of water in oil emulsion with Mycobacterium tuberculosis components? So it is complete Frounce adjuvant. 2. Karyogram of an individual show presence of 45 chromosome, 44 plus X, and one sex chromosome is missing. So X0. The individual has a female appearance and dwarfism. Which of the following is the most probable condition associated with the individual? That is Turner's syndrome. Answer is option 2. Match the components of list 1 with those in list 2. Idiopathic thrombocytopenic purpura ITP is platelets. Hashimoto's disease is thyroid. Celiac disease is gut, and Huntington's disease is brain. So the answer here is option 3, A4, B1, C2 and D3. Then which family does HIV belong to? It belongs to Retroviridae. Answer is option 1. Kuru disease in human is caused by prions. Answer is option 3. Which statement is true for pathogenicity island? These are large segments of bacterial genome encoding virulence factors. 1. Answer is option 1, that is true. Then, which one of the following diseases can be treated with dopamine producing neurons generated from stem cells? So it is Parkinson's disease. Answer is option 1. Protein A, which has strong affinity for Fc region of immunoglobulin, is extracted from. It is extracted from Staphylococcus aureus. Answer is option 2. Which one of the following diseases is caused due to a point mutation in the coding region of the associated gene? Alpha thalassemia, beta thalassemia, sickle cell anemia, hemolytic anemia. It is sickle cell anemia, which is a point mutation. Answer is option 3. So coming to the next question. Double pain sensation that is occasionally felt following painful stimulation of the skin is due to. So double pain sensation is two sequential pain sensations in short time interval. The first one is immediately after the damage and it is followed several seconds later with additional pain sensation. So it is due to the presence of dual pain pathways. Answer is option 2. Then next one. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion. It is essential that the animal cell cultures be maintained in antibiotic-free conditions, otherwise cryptic contaminations will persist. The constant use of antibiotics favor development of chronic contamination. Many of sorry many many organisms are inhibited but not killed by antibiotics, which may resurface when the conditions are favorable. In the light of the above statement, choose the correct answer from the options below. So here both A and R are true and R is the correct explanation of A. Answer is option 1. Next one. Match the components of list 1 with list 2. Hydrocortisone, glia, glioma, retinoids, endothelium, prolactin, mammary epithelium, interferon gamma, neuroblastoma. So the answer here is option 2, A3, B2, C4 and D1. The next one, mosaic spheroids, bystander effects, microcarrier matrix, 3D growth environment, organoids, 3D aggregate of cells, rotary cell culture system, microgravity cell growth environment. So the answer here is option 3, that is A3, B4, C1, D2. What is the role of macrophage activating factor in an animal cell culture media? It is antiviral. Answer is option 2. Common indicators of water pollution with enteropathogens are following except Bacillus species. Answer is option 4. The first US patent for a GM organism was awarded to Dr. A. Ananda Chakravarti for his work on one of the following. That is Pseudomonas engineered to degrade petroleum. Answer is option 1. The suitable method for treatment of municipal waste water and aqueous hazardous waste which has less than 1% of suspended solids is? Answer is activated sludge process. Coming to the next question. Nitrification during nitrogen cycle is the production of. So nitrification is a microbial process by which reduced nitrogen compounds, that is ammonia, are sequentially oxidized to nitrite and then nitrate. So answer is option 1, nitrates. The next one. Humans mainly obtain DHA and EPA. DHA is docosahexaenoic acid, a type of omega-3 fatty acid, and EPA is icosapentaenoic acid, which is also an omega-3 fatty acid. So by consuming fish, whereas fish in turn obtain PFAS from microalgae. Statement 2. Microalgae derived DHA and EPA can be used as supplement for people who do not consume fish and seafood. So both the statements 1 and 2 are correct. Answer is option 1. Then match the following. Legume Azorhizobium, Azolla anabana, sugarcane, Acetobacter actinorhizal frankia. So answer is option 3, that is A2, B3, C4 and D1. Then match marine enzyme with their source. So chitinolytic enzymes, digestive tracts of fish, shellfish, squid liver, octopus saliva. Gastric proteases, fish viscera from fishery sources. Polyphenol oxidases, crustaceans. Serine and cystine proteases. Pyloric ceca, pancreatic tissues, intestine of sardine, cod and salmon. So answer is option 1, A1, B3, C4, D2. Then next one. Given below are two statements. Statement 1. Xenobiotic pollutants are biomagnified and accumulate in marine organisms. Statement 2. Pollutants can be quantified in tissue samples from key marine animals living in the environment where pollution monitoring is in place. So both the biomagnification, it refers to the accumulation of toxic substances in the food chain. So xenobiotic pollutants, they are biomagnified and accumulate in microorganisms, and pollutants can be quantified in tissue samples from key marine animals living in the environment where pollution monitoring is in place. So this both the statements 1 and 2 are correct. Answer is option 1. Brine shrimp assay involves, so brine shrimp lethality bioassay is a simple high throughput cytotoxicity test of bioactive chemical. It is based on the killing ability of test compounds on a simple zoological organism, that is brine shrimp. So answer here is testing the toxicity of anti-cancer molecules using eggs of brine shrimp. Suppose you have got some bioactive chemical, that is phytochemical compound. You have to conduct bio shrimp lethality assay to know the toxicity. And it is based on killing ability of the test compound on a simple zoological organism, that is brine shrimp. Strong adhesives can be prepared using the constituents of one of the following. Answer is option 3, that is bices generated by sea mussel. Then match the components of list 1 with list 2. Rhodopsin, vitamin A, tocopherol, vitamin E, isoflavonoids, soya bean, ascorbic acid, vitamin C. That was very easy one. So it is answer is option 3, that is A2, B3, C4 and D1. Which one of the following is not a fermented food? Sauerkraut, cheese, salami, milk cream. Milk cream is not a fermented food. Next one, given below are two statements. One is labeled as A and the other is labeled as R. Assertion. Within a few hours after an animal is killed, rigor mortis set in with the contraction of muscle fibers and an increasing toughness of the meat. The loss of glycogen and disappearance of ATP from the muscle are observed in freshly killed animals. In the above statement, choose the correct answer. So both A and R are true and R is the correct explanation of A. Answer is option 1. Which one of the following methods of controlling microbial contamination is the least preferred in food processing? So it is dry heat. Answer is option 3. Spirulina is considered as a superfood for human consumption because it contains all essential all essential amino acid, vitamins and fatty acids. It does not contain carbohydrate and lipids. So answer is option 3. So that's it. Thank you for watching my channel. Please do like, share and subscribe. And one more thing I would like to suggest is that those who are appearing for CSIR, DBT, GATE exams can also refer PhD entrance exam question papers. I have uploaded some in my channel. You can go through them. They are also very useful.